How's everybody doing?
Amazing.
Amazing? Everybody's doing amazing. You're speaking for everybody here, right? Okay, so we're not too far from finishing up regulation of enzyme activity. When I finished last time, I had started, at least introduced the topic of blood clotting. And as we will see, blood clotting is another system that uses zymogens. It is a system that, in addition to using zymogens, uses an interesting scheme or an interesting strategy that we will see later, and it's what I call a cascade. And the cascade system is used in other enzyme control systems. And the beauty of a cascade system is that you can mobilize an effect very, very rapidly. Okay? What does that mean? So if I think about the Cascades, the Cascade Mountain Range, and I go climbing the Cascades and I get up to a high point, the higher I get up, the more I see that the cascading waterfalls get smaller because I've got a smaller source of water. As I go down the mountains, the further I go down the mountains, the waterfalls and everything get bigger because the streams start coalescing together. That cascading system, one stimulating another, stimulating another, stimulating another, is a very, very effective way to make things happen very rapidly. Okay? As I said, we'll see another example of it, but this shows a very involved system for blood clotting. I'm not going to take you all the way through it. In fact, I'm only going to emphasize a couple major points in it. But suffice it to say that a cascading system, if we think about a signal, let's say it's a damaged tissue or a damaged blood vessel in some way, that signal has to be amplified. And the reason it has to be amplified is because, as I said at the end of the lecture last time, we really need to stop that blood flow before we lose too much blood. If we don't, then the person will bleed to death. So we have to have a system that works very rapidly and is very effective. All right? So a small signal here, if this is an enzyme, this enzyme activates another enzyme, and this enzyme in turn activates a bunch more enzymes, and this bunch of enzymes activates an even bigger bunch of enzymes, and at every step along the cascade, the signal gets bigger and bigger and bigger. The beauty of this is that it happens very rapidly. It's happening in our bloodstream. And so that the main thing that we have to do in order to protect ourselves via a clotting mechanism is to make sure that we have plenty of zymogens in our bloodstream, things that aren't yet clottable, but that can be very quickly turned into a clot. Now, that's the good side. The bad side is that the same thing. We have a bloodstream that's full of things that can form a clot. And so if the system screws up, then we can very rapidly form a clot and kill ourselves if we have the clot forming in the wrong place. All right? So there's a yin and a yang to blood clotting. We're going to focus mostly on the good side, I guess the yin. I was going to say good, but on the positive side of the blood clotting, which is the forming of the clot. And I'll also talk about how we dissolve a clot. So just like, as I said before, if the body has a way of turning a system on, it's also got to have a way of turning a system off. If it makes blood clots, it's got to be able to dissolve those blood clots as well. And we'll talk about both of those. Okay. So like I said, I'm not going to go through this pathway in detail, and no, I'm not going to ask you to regurgitate this pathway to me. All right? The important aspects of this pathway are actually down here for our purposes. Okay? We don't really care much of up here. For our purposes, we're going to focus on what's happening down here. Okay? Now, as we start with things on the left, or things, I shouldn't say things on the left, but things that are coated in this sort of pinkish color, these are inactive zymogens, or inactive factors. So when I see, for example, prothrombin, prothrombin is a zymogen. It is inactive. It is incapable of acting. By the way, most of these guys on here are proteases. One protease activating another protease, activating another protease, activating another protease, and that last protease is going, which happens to be thrombin, is going to convert the clotting material from an inactive form to an active form, meaning that the clotting material will start to make a polymer. So once this guy right here gets converted to fibrin, fibrin is a self-assembling polymer. That's very important. It's a self-assembling polymer. In the absence of this activation, fibrin is floating through your bloodstream all the time, or fibrinogen is floating through your bloodstream all the time, doing nothing. That's what you want it to do. You don't want it to clot unless you've got an issue. Well, this raises a couple of concerns. One is we want clotting to occur in a specific place. We don't want it occurring randomly in our body. And our body, I'll show you one way, has a way of knowing where to put that clot. There are several things in place that help to do that, but I'm going to tell you about one of them at the molecular level. So our focus for right now is going to be on the prothrombin to thrombin, and the thrombin catalyzing the conversion of fibrinogen to fibrin. Okay? All right. So let's consider that. Where was I at here? There we go. First of all, we have to consider the structure of fibrinogen. So fibrinogen is the inactive polymerizing material. When it gets activated, then it will form polymers. Well, it's kept inactive by the action of these two things here, this labeled with its capital B and this guy down here with its capital A. These we can think of as knobs that basically stop the polymerization process. During the polymerization of fibrin, what we will see is these knobs get clipped off, and they yield ends that can link into these things that we see here. Okay? You see this little hole? The B will fit into that little hole. So I can take one fibrin molecule that's got a B and stick it into the hole of another one that's got this. The A's will stick into the gammas. Okay? So what thrombin is doing is it's clipping off the knobs. It clips this guy off, it clips these guys off, and now we've got some ends that the pieces can start sticking together. The tinker toys, as it were, can start building themselves into a bigger structure. All right. So that actually occurs in this mechanism that you see here. Okay? In this case, the alphas are linked up with the gammas, as we can see here. Okay? The alphas and the gammas have been stuck together. We don't see the betas going into the B's, into the beta structures, and the reason is because that goes in the third dimension. We can imagine that this polymer is going to stick back out towards us, right? So we have this guy sticking in here, this guy sticking in here, this guy sticking, let's say, in here, and now we get a three-dimensional structure. So coming back out at us, we've got all these things tied together. Now that's, what's amazing is A, it happens very rapidly. Okay? It happens very, very rapidly. And second, it's a pretty good structure, but it's not a perfect structure. What does that mean? Well, what you see on the screen is a sort of a two-dimensional display of what we call a soft clot. A soft clot, why do we call it a soft clot? Well, it's the very first thing that forms when there's been damage, or there's been a cut, and you're losing blood. The very first thing that happens is what's called a soft clot. And the reason we call it a soft clot is that these interactions are not, underline not, covalent. These are hydrogen bonds. It's soft because, yes, it helps to get all the pieces together, but it's not very sturdy. It doesn't hold things real well. We can imagine we get a few hundred or a few thousand of these hydrogen bonds. It's really going to start to add up to a reasonable structure. But for a good protection, we want to have what's called a hard clot. Okay? To get a hard clot, we have to make covalent bonds. All right? So what you see in terms of that initial polymerization reaction only makes hydrogen bonds. It does not make covalent bonds. The covalent bonds require action of another enzyme. Okay? The other enzyme is known as a transglutaminase. And it catalyzes a reaction like this. The side chain of a glutamine and the side chain of a lysine can be joined together to make a crosslink. This is a covalent bond. Now, this is not happening in those little knob structures. This is happening just between the strands when they get adjacent to each other. If there is a lysine next to a glutamine, this transglutaminase will join these bonds together. When we make these covalent bonds, we've converted a soft clot into a hard clot. Okay? We've converted a soft clot into a hard clot. If you watch a scab on your hand, you'll notice that when it first forms, it's different than what it looks like a couple of hours later. Okay? It goes from being literally soft feeling to being hard. The scab does that. Okay? Now, as I said, the remarkable thing is this happens in the order of minutes, and this happens at the place you want it. It's rare that you're forming clots in places in your body where you don't want to have it. Okay? And it's watertight. Those are really remarkable features of blood clotting. Okay. Well, how do we know, how does the body know where to make that? How does the body know where to do it? Okay? One of the ways in which the body uses information about how to do it is by a modification to prothrombin. Prothrombin. Remember, prothrombin is the zymogen form of thrombin. It's the inactive form of thrombin. But the body has a way of collecting prothrombin at the site of the wound. It has a way of collecting prothrombin at the site of the wound. I need to tell you about that. Okay? So that happens as a result of action of prothrombin. Let's see, let's go here. Here's what I want to show you. Okay. In order for prothrombin to get gathered at the site of the wound, it has to be modified. Okay? So prothrombin gets modified by vitamin K. Vitamin K is known as the clotting vitamin. All right? Vitamin K is required by an enzyme that puts an extra carboxyl group on the side chain of glutamate. So prothrombin has several glutamates. This enzyme that uses vitamin K grabs a hold of prothrombin, it grabs a hold of vitamin K, and it puts additional carboxyl group on the side chain of glutamate. Okay? Here's a regular glutamate side chain. Here's the addition of a new carboxyl group on it. Okay? So in black, you see the regular, going up here, the regular side chain of glutamate. All right? And now this guy has had an extra carboxyl group added to it. Why is that important? Well, it turns out that when that extra carboxyl group gets added, prothrombin can all of a sudden bind calcium very, very well. It can bind calcium very, very well. With only one carboxyl group, prothrombin doesn't grab calcium worth a darn. All right? But with two carboxyl groups, they sort of gang up on calcium and hang on to it. Okay? One doesn't do it. Two are positioned perfectly to bind calcium. Calcium is charged +2. Each carboxyl group is charged -1. They form a very nice bond. Why is that important? Well, at the site of a wound, we've got cutting, we've ruptured open cells, and we've exposed a bunch of calcium. Okay? So right at that site of the wound, we're going to have an abundance of calcium there, and that calcium is going to attract prothrombin. Prothrombin will be concentrated at the site of the wound. Now, prothrombin sits there and waits for all of the other zymogens to get activated, and finally it gets activated, making thrombin. And what's thrombin going to do? Well, thrombin is going to convert fibrinogen to fibrin, and right there at the site of the wound, that polymer is going to form. So there's other systems the body uses, but one is this one. It's vitamin K dependent. It's why we have to have vitamin K to have efficient blood clotting. Okay? Yes?
So when you get a thrombocyte, what is a thrombocyte?
Okay, so there's many causes of stroke, but one of the most common causes of stroke can be the formation of a clot in a place that would stop blood flow to a vital organ like the heart or the brain. Okay? And yes, those do happen, and those are a problem. And to prevent the formation of clots in places where you don't want them, people are given what are called blood thinners. And I'm going to talk about that in just a second. Okay? Yeah, Liz. Yep.
What goes wrong?
If you're a hemophiliac, what goes wrong? There's several places in this scheme where you can be lacking an enzyme genetically. So if you're lacking a critical enzyme, and there's several places where this can happen, if you're lacking a critical enzyme in that activation pathway, you may not be able to convert zymogens, and that's going to stop the whole cascade, and you're literally going to bleed to death if you don't have that factor. But there's several places where that can happen. Okay, so a very interesting phenomenon, a very important phenomenon. It has a molecular basis. When I talk about vitamin K, this is what vitamin K looks like. Okay? Vitamin K is needed by that enzyme that puts the carboxyl groups on the side chains of glutamate of a prothrombin. All right? Blood thinners, okay, the things that people refer to as blood thinners, resemble vitamin K. And the enzyme binds those molecules, and when it binds those molecules, it cannot put a carboxyl group on the side chain of prothrombins. Would you describe these guys as competitive or non-competitive? These are competitive. They resemble, in some way, vitamin K. They're competing for the same site. Warfarin is also known as rat poison. That was the original use of warfarin. Oh my God, if I poison rats, am I going to get blood all over my house? No. Do you know why? Internal bleeding, yeah. So when you really thin the blood a lot, what happens is the most common thing that happens is the slightest bruise can kill you. So people who get put on blood thinners, okay, they have to do what we call titrate the thinner. We don't want to give them too much thinner because we will kill them if we thin their blood too much. They will bleed to death internally. So a physician who's giving a person thinners will measure what's the clotting ability of this person. You're trying to lower it but not stop it because you don't want to completely stop it or you're going to kill the person. Okay? Warfarin and coumarin both are very effective in this respect. They both do reduce the clotting ability, and people, for example, who've had a stroke or have other problems. I have relatives who have phlebitis. Phlebitis is a clotting disorder in the legs, and they get put on blood thinners to keep them from forming these clots in their legs. And again, they have to balance the right amount so they don't give them too much. At the same time, they want to stop the clotting as much as they can. I'm sorry?
A deep vein thrombosis?
A deep vein thrombosis, huh? Yep. Okay. All right, let's see here. What else am I going to say? All right, so that's how we form clots. And that's a fairly cursory look of how we form clots. I also want to say a word about how we get rid of clots. Because as I said, the body has to not only make things, if it has a switch that turns something on, it has to have a switch to turn something off. And so how does it get rid of clots that it forms? Well, it turns out our body has an enzyme that does this very, very well. The enzyme that dissolves blood clots is also a protease, and it's known as plasmin, P-L-A-S-M-I-N. Plasmin. And plasmin is present in the bloodstream, not as plasmin, but as plasminogen, because we want to have that available so that we can activate it when we want to dissolve the clot and make that. All right? Now, how do we activate plasminogen? Well, that's activated by another enzyme known as TPA, which also stands for tissue plasminogen activator. Tissue plasminogen activator is also a protease. We see a lot of proteases involved here. And the effect of this protein is remarkable. Now, TPA has the historical note that it was the first genetically engineered protein that was made available for human use. Okay? It was actually the first protein that Genentech, if you've heard of Genentech, that was the first protein. They were the first one to get that approved. TPA is a very powerful molecule. It needs some activation as well, and we're not going to talk about how all that occurs. But TPA, basically, if you give TPA at the site of a blood clot, it will activate plasminogen at that site and effectively break down a clot. Now, it's not given routinely because you could imagine you would have some problems turning this on, like giving too much blood thinner. But at the site of a clot, this guy can convert a plugged artery in the heart to complete flow through in a matter of minutes. Okay? So for a person who has a blocked artery because of a clot, TPA can be a lifesaver. In some cases, TPA is actually given to people after they've had a stroke in hopes that if there are small clots in the brain or something, that it can be dissolved very quickly and readily, that they can actually alleviate the effects of a stroke, and in many cases that actually can have a very positive effect. As I said, it's used very carefully because, again, it's a very, very powerful substance, and we don't want to be just indiscriminately breaking down clots that might otherwise be protecting us. Yes, sir?
I know that in situations like you're describing, they kind of colloquially refer to that as a clot buster.
Mm-hmm.
What portion of that is TPA, or are there other components that would generally use?
Yeah. His question is, when you hear about the term clot buster, does that refer to TPA or other things? And there are other things that can be used as well, but TPA, the term clot buster is just a generic term. TPA is definitely a clot buster, yes. Yeah. Okay, yeah, back there. Ooh, boy, that's a very good question. How long does TPA remain active in the bloodstream? And I don't honestly know the answer to that question. Don't know. Okay, so that's the activation, the inactivation of blood clotting. It's a pretty phenomenal process, I think. And if there are no other questions, I'll move forward to carbohydrates. One other question back here. Yeah, Elliot.
What was which enzyme catalyzes the vitamin K and
Yeah, his question is which enzyme catalyzes the carboxylation of prothrombin. I didn't give you the name of that, so you're not responsible for that, no. Vitamin K is a cofactor for that enzyme. Okay. All right. Well, we turn our attention now to a subject that most students tend to like because this subject is something they've covered before in organic chemistry. This is structure of carbohydrates, and it's almost all focused on structure. All right. So we'll say a lot about the structure of carbohydrate. There's a lot of terms that are here, and it's very straightforward kind of stuff. So I'm going to go through it sort of quickly, but also hopefully not too fast to run over you with that. We talk about carbohydrates. Carbohydrates are obviously important molecules for us. Carbohydrates are one of our main sources of energy. They are, in fact, our primary source of quick energy. All right? Carbohydrates include sugars. They include polymers of sugars. And they also include modified forms of those sugars. Okay? The term carbohydrate actually tells us what the structure of the molecule is. Carbo referring to carbon, hydrate referring to water. All right? Carbohydrate is basically the structure of these molecules. Let's, for example, look at the structure of glucose. The structure of glucose is C6H12O6. You don't need to write that down. All right? But I could easily write that C, okay, X, H2O, X. Right? In this case, the X is 6. That's a 6-carbon sugar. What if I had a 4-carbon sugar? Well, that'd be C4H8O4. So it's a hydrate of carbon. That's what a carbohydrate is. Water to carbon. Okay? Probably never thought about that before. All right. Well, the first term I want to introduce you with respect to carbohydrates, and by the way, we also use the term carbohydrate, we use the term saccharide. Saccharides are the same as carbohydrates. S-A-C-C-H-A-R-I-D-E. Saccharide. A saccharide literally means, and I think it's Latin, sweet taste. Okay? Sweet taste. So carbohydrate, saccharide, same thing. Well, let's look at a couple of structures of very simple carbohydrates or saccharides. Okay? These are 3-carbon molecules. In this case, we would have C3H6O3. We notice that they are similar in structure but not identical. We see, first of all, that this guy is a ketone, and these guys over here are aldehydes. Okay? Ketones versus aldehydes, right? And if you look at the two on the right, they are both aldehydes, but they are slightly different in their three-dimensional configuration. We remember that a carbon that has 4 different groups attached to it can have those groups attached in three-dimensional space in 2 different ways. And here's where you're going to like biochemistry, because biochemists are very simple people. We like to think of the terms D and L to describe those. And you'll see this is a great simplification in terms of describing their overall structure. The ketone doesn't have, in this case, doesn't have an asymmetric carbon. There's no carbon that has 4 different groups attached to it. So there's only one form of this 3-carbon ketone. A carbohydrate that has a ketone bond in it is called a ketose, K-E-T-O-S-E. Whenever you see the letters OSE on the end of a name, we're talking about a carbohydrate. A ketose is a general name for a carbohydrate that has ketones. Fructose is a specific ketose. And I'll show you the structure of that. Okay? On the other hand, if instead of having a ketone bond in it, that the carbohydrate has an aldehyde bond in it, that structure is known as an aldose, A-L-D-O-S-E. And again, that's a general term for a carbohydrate that has an aldehyde bond in it. Okay. We can further delineate the names of these guys by describing the numbers of carbons that they contain. The guys I just showed you are 3-carbon molecules. They're known as trioses. I could describe them as an aldotriose or a ketotriose, depending upon whether they had an aldehyde or a ketone bond in them. If they have 4 carbons, they're known as a tetrose, 5 a pentose, 6 a hexose, 7 a heptose, 8 an octose. We don't generally see carbohydrates with single units containing more than 8 carbons. But we will see polymers of some of them that have 6. Okay? Now, when we look at the different structures of carbohydrates, we see that there are a variety of names that can be used to describe these. Let's start down here. Okay? This guy down here shows those two aldoses that I showed you before. One is known as glyceraldehyde, D-glyceraldehyde. The other is known as L-glyceraldehyde. You notice the structure, C3H6O3. Okay? They are mirror images of each other because, again, that relates to the three-dimensional arrangement of those those substituents on the asymmetric carbon. We draw them in simple terms. We don't even draw the three-dimensional. We can draw it like this, where we take the asymmetric carbon and we put the hydroxyl on the right side versus putting the hydroxyl on the left side. In general, when we look at the structure of carbohydrates and we decide if it's D or L, we look at the next to last carbon. If the OH is on the left side of the next to last carbon, it's an L sugar. If it's on the right side of the next to last carbon, that is from the bottom, okay, it's D sugar. Okay? If we orient the OH on the left side of the next to last carbon from the bottom, it's an L sugar. If we orient it on the right side of the next to last carbon from the bottom, it's a D sugar. 2 carbohydrates that are mirror images of each other are called enantiomers. Yes?
Are there biases in, like you talked about with proteins, with D or L?
Yeah, are there biases? Do we see carbohydrates being in one versus the other? We do tend to see many more in the D form than in the L form. Mm-hmm. Yes, we do. But it's not as strong as we see with amino acids and other things. But D is very strongly favored. Okay, so 2 molecules are enantiomers if they are mirror images of each other. These guys are mirror images of each other. Okay? Now, I don't even use this term constitutional isomers. I don't like that term. So we're not even going to hold you responsible for that. What is a stereoisomer? Okay? Stereoisomers are molecules that have the same general structure. That is, they're both C6H12O6, all right? They're both aldoses, but they're not mirror images. Look at this, okay? This guy is not a mirror image of this one. They are stereoisomers of each other. And another term is used, it's not on here, oh, actually it is right here. They are called diastereomers. D-I-A-S-T-E-R-E-O-I-S-O-M-E-R-S. So stereoisomers will include enantiomers. They will also include diastereomers. Now, notice what I said had to be for a diastereomer. They had to be the same kind of sugar. In this case, they had to both be aldoses. They had to have the same number of carbons, but they're not mirror images of each other. All right. Yes, sir.
How come the D aldose has 7 oxygen?
I'm sorry?
The D aldose?
Has 7 oxygens instead of 6.
1, 2. Oh, that's a very good question. That's actually incorrect. I didn't even notice that. Obviously, the book didn't either. But no, that should only be an H. Yeah.
Okay.
Yeah. Good eyes. Wow. I've stared at this, and I never noticed that before. When I was working on a textbook, I was a co-author on a textbook about 10 years ago, and I was on the 3rd edition of the textbook, and so I was reading all the things that the other authors were writing and so forth, and we used a lot of the same figures in our textbook that had been used in the previous edition of the textbook. And so I look at this one figure, and I said, "This is ridiculous. It's a carbon that's got 5 bonds." Okay? And so I go to my co-authors and I said, "This carbon has 5 bonds." And they said, "Oh my God, it's been in the last 2 editions and nobody's ever noticed it." So you found 5 bonds. So you found an extra oxygen. Good. We should contact. We've seen other errors in this edition of the textbook, so that's kind of bad. All right. This is the first time this figure has been used in this edition of the textbook. So we didn't used to have this figure, and I kind of like this figure. And there's my little. Okay. Now, 2 other terms, all right? So, stereoisomers include enantiomers, and they include diastereomers, okay? Diastereomers include epimers, and epimers are 2 sugars that, again, have the same kind. They're both, in this case, aldoses. They have the same number of carbons. They only differ in configuration by 1 hydroxyl, all right? So if we look at this guy, on the left, on the left, on the right, on the right, on the right, on the right. The only place they differ is right here. These 2 guys are epimers of each other. They're not mirror images, okay? They're not mirror images of each other. They're epimers. They only differ in the configuration of carbon number 2. Last, anomers are also diastereomers, and anomers arise from the different configuration that comes upon cyclization. Now, I haven't said anything about cyclization, so I'm going to show you that in a minute, but I'm going to introduce the term to you right now, and then I'll come back and show that to you in more detail in a minute. Okay, so you should know what an enantiomer is, you should know what a diastereomer is, you should know what an epimer is, you should know what an anomer is, you should know what a stereoisomer is. Okay. Just some basic terms of carbohydrates. All right, here are some common monosaccharides that we see. We call them monosaccharides because they're not polymerized. They're only existing as a single unit. Glucose is a monosaccharide. Fructose is a monosaccharide. Galactose is a monosaccharide. Ribose is a monosaccharide. Deoxyribose actually is an oddball because it's lacking in oxygen. That's why we have the deoxy part. Okay? We show it to you here because obviously this is a very important constituent of DNA. That's what gives DNA the D part of its name. Okay? Now, when we start looking at sugars, carbohydrates here, we start thinking, okay, what's Kevin going to make us know all these structures? Well, there are hundreds of possible carbohydrate structures. That pained look says, I really don't want to know that, right? I'm not going to make you know all those, but I will make you know the structures of the important ones. And the reason I make you do that is you're going to need to know them in other classes, all right? So you should know the straight chain and the ringed structure forms of glucose, galactose, fructose, and ribose. Ring and straight chain, glucose, galactose, fructose, ribose. Now, these actually are quite common. They're quite similar to each other. Let's look at glucose and fructose, for example. The only difference between them is glucose is an aldose and fructose is a ketose. Because if we look at the configuration of the OH groups, all right, this one is lacking an OH on position 2, so we go to position 3, it's on the left, position 4 on the right, position 4 on the right. Glucose and fructose are identical, except for whether they're ketose or aldose. Okay? Galactose is an epimer of glucose. It's an epimer of glucose. The only place that galactose differs in configuration from glucose is right here. These are very easy to learn in terms of structure. I learned glucose is right, left, right, right. Right, left, right, right. I can always draw glucose, and then I remember that this guy is going to be identical. I remember that galactose is going to differ at carbon number 4. 1, 2, 3, 4. There's the difference. Okay, so you can put these to memory pretty easily. Now, you learned in organic chemistry, I trust, that these 6 carbon or 5 carbon rings have a geometry such that they can actually come back around and interact with each other to make ring structures. The ring structures are named according to their resemblance to a couple of molecules. Puran is a molecule that looks like what you see on the top. It has 6 carbons, and it has 5 carbons, and it contains an oxygen. Furan has 4 carbons and contains an oxygen. This is a 6-membered ring. This is a 5-membered ring. We use these names as our way of describing sugars. Sugars that form 6-membered rings we refer to as pyranoses. Notice I said 6-membered rings. 6-membered rings have 5 carbons in them. Okay? Sugars that form 5-member rings are known as furanoses. 4 of those are in there. Okay? Can a 6-carbon sugar form a pyranose? Yes. Can a 6-carbon sugar form a furanose? Yes. All right? We're only counting the number of carbons in the ring. Other carbons can be sticking off, as we will see. Okay. So how do these form? Well, aldoses form a structure known as a hemiacetal. A hemiacetal arises by reacting an aldehyde with an alcohol. Okay? An example would be glucose can make a hemiacetal structure because glucose is an aldose. A hemiketal arises from taking a ketone and reacting it with an alcohol to make a hemiketal. And fructose is a ketose, so it can form a hemiketal. Let's watch this cyclization process happen. The cyclization process happens, as you can see right here. Here's the 6-membered glucose. The 6-membered glucose has a geometry such that this hydroxyl group on carbon number 5 right here can get very close to that aldehyde group on carbon number 1. When it does, it can make a ring structure. And it turns out it can make 2 ring structures because what's happening is this guy, which only had 3 members on it, now is going to have 4 members on it. We don't have the double bonded oxygen anymore. If it forms in this configuration such that the OH is down, we refer to that configuration as the alpha configuration. If it forms such that the hydroxyl is up, we refer to it as the beta. Now, at this point I can tell you these 2 guys we see here on the right are anomers. Their only difference is in whether they are alpha or beta. Everything else is the same. Okay? So we can have alpha D-glucopyranose, we can have beta D-glucopyranose. Those 2 will be anomers. But if I had alpha D galactopyranose and beta-D-glucopyranose, they would not be anomers because they would have other differences. Right? So anomers can only differ in the configuration of the anomeric carbon. And by the way, the anomeric carbon will always be the carbon that had the double-bonded oxygen. I'll repeat that. The anomeric carbon will always be the carbon that had the double-bonded oxygen. That's true whether it's an aldose or whether it's a ketose. Okay, so we have a glucopyranose here. We have a glucopyranose here, alpha versus beta. Yes, Shannon.
So is an anomer also an epimer?
Is an anomer also an epimer? Technically it is, but we don't tend to use that term for that. We use epimers as other than the anomeric carbon. Okay? All right. When we have a ketose, like fructose, what happens? Well, looky here. Here is our anomeric carbon. There's a double-bonded oxygen. Here's carbon number 5. We see this same sort of intermediate structure forming here. And voila. We have, in this case, alpha-D-fructofuranose. Alpha meaning the hydroxyl is down. Notice it's the hydroxyl that determines the position of alpha or beta, not the carbon that's there. All right? Can we have a beta-D-fructofuranose? Yes, we can. Okay? They just haven't, your book has gotten lazy and they haven't drawn it. If we had the beta, we would have the same structure, so we'd have the hydroxyl up, and we would have the CH2OH down. Now, notice, this is a 5-membered ring. It has 6 carbons. Carbon number 1, 2, 3, 4, 5, 6. If you want to spare yourself grief on the exam, always number your carbons. You'll always bail yourself out. Don't forget to number your carbons, because the most common things I'll say, what's the structure of fructose? Oh, 5-membered ring, 1, 2, 3, 4, 5, I've got it. Well, you know that fructose is a 6-membered, is a 6-carbon molecule. It's a hexose, right? Okay? So don't forget to number your carbons. That will always bail you out. Okay, now, an important thing about these structures is that they are reversible. They are reversible. In solution, which is the most common way that we have glucose, these molecules can go back and forth from one structure to the other structure quite readily. Okay? They can go from one structure to the other structure quite readily. And it turns out that there's a little bit of steric hindrance for the beta form. I'll show you that in just a little bit. And so we tend not to see as many of these in the beta form as we see in the alpha form. But nonetheless, we do see some in the beta form. In solution, it's going back here, over here, back here, over here. One of the criteria for the reversibility of this process okay, is the ability of these guys to go back to the straight chain. If they can't go back to the straight chain, they can't flip. These guys can flip. Well, what stops it from going back to the straight chain? If I modify the hydroxyl. If I modify the hydroxyl, that process is not reversible, and it will be stuck in that configuration. It will stay. So let's say I put a methyl group in place of that hydrogen right there. If I put a methyl group right there, it can't go back. It will stay stuck in the alpha configuration in this case. Let's say I put a nitrate right there. Same problem. It's not going to go back. It's going to stay, in this case, in the beta configuration. So if I do anything to that anomeric hydroxyl, and notice it's the anomeric hydroxyl, I will lock it in whatever configuration it happens to be in.
So they just switch between alpha and beta. They don't switch between straight and right.
Well, they can make straight. Yes, they can make straight. Yeah, they have to be able to go back to straight and back to here. Yeah. Okay? Okay. So, don't waste your time on this slide. Okay? Since I've shown it, I'll tell you, okay? I'm going to show it to you. You're not responsible what's here. All right? This is simply showing you that, yes, these guys can make 6-membered rings, just like glucose can make 6-membered rings. It's not the most common form we find fructose in, and I think it just adds another level of memorization that you don't really need, okay? So we're not going to worry about the 6-membered rings of fructose, okay? Okay. Now, these are the ring structures of sugars that we find very commonly. Okay? Yes, you're responsible for ribose. Yes, you're responsible for glucose. Yes, you're responsible for fructose. And yes, you're responsible for galactose, alpha and betas. Okay? And again, these are very, very similar to each other, but don't forget to number your carbons or you'll get lost. Look at fructose. Whoa, those aren't identical. Yes, they are. There's carbon 1, 2, 3. There's carbon 1, 2, 3. Hydroxyl up on 3, hydroxyl up on 3. If you don't number your carbons, you will get lost. Okay? Yes?
So the ribose ring doesn't have an alpha or beta indication. Does it only form one?
It actually should have that. If I were to say what configuration would that be, what would you say? That's beta. That's beta. They do have alpha and beta. What we see in nucleotides is it's always in the beta. I think that's why they haven't drawn it or given that designation. But you're right, it should have that designation on there. So this would be the beta D-ribose right here. Okay? Good. You guys have good eyes today. Okay, let's see. Let's say a word about steric hindrance. I've mentioned it. We talked about steric hindrance earlier in the term, and steric hindrance relates to nuclei or electron clouds that get too close to each other. And we saw that there's a tremendous amount of energy, okay, that oppose putting things too close together. This is a schematic way of looking at a sugar that has a couple of groups that are kind of butting heads with each other. And we could imagine that if there was a way for the sugar to avoid butting heads, it probably would do that. And in the case of an anomeric carbon, it's actually fairly readily able to change that. Okay? This shows glucose in the beta configuration. Okay? The beta configuration, we can see that this guy over here has a hydroxyl that is sort of interacting with this CH2OH on carbon number 6. They're too close to each other. We describe the structure that this guy has as a boat, because if we trace the path of the carbons, okay, the carbons look like this. There, and then down, and then across like this. It looks like a boat. All right? The same beta, this is beta, this is beta, they're both betas, can twist bonds and rearrange itself so that that interaction does not occur. This is a beta form like this is a beta form, but this is a beta that has a different conformation, and it's flipped itself so that hydroxyl, which was in the way up here, is flipped down over here. Okay? And I shouldn't say down, it's flipped equatorially instead of being flipped up. All right? Now, that configuration is called the chair because it has a sort of configuration of a chaise lounge. There's the back, there's the butt end part, and there's where you put your feet. You have a question?
Yeah, over here, that hydroxyl that's pointing not quite axial up, shouldn't that have very little steric interaction with the CH2OH because it's equatorial going into the plane?
These two guys do interact, actually, right here. Yeah. So there is interaction. There's much more interaction here than there is here. And so this is favored of the two structures. The chair form is favored over the boat form because of that. So if you compare this to this, this has much more interaction than this one does. Yes, Connie?
When you say the chair form is favored, do you mean in all cases or just
In this particular case. But in general, when you can arrange things away from each other, you're going to be better off. But I'm just illustrating this as one example to show you how a boat versus a chair form might be a favor for structure. Yes, sir?
Will we need to put these in chair or boat form on the example?
Well, we need to draw a chair or boat form on the exam and get all these axially and so forth, and the answer is no. I think that's kind of busy work. But I think you should certainly know what a chair form is. And I think you should know what a boat form is and why one versus the other might be favored. Question back here?
So maybe I missed it. How do you determine whether it's alpha or beta?
Okay, so the alpha has the hydroxyl down as we draw it, and the beta has the hydroxyl up. Okay, that's a good place to stop. I hear the rustling. I'll see you guys on Friday. Yes, sir.